For our Distinguished Speaker Series, Jon-Kar from the University of Michigan. Jon-Kar originally hails from Spain, which is where he did his medical training. He spent a bit of time at UNC, which must cause some issues because UNC in Michigan, but where he started his training in neuroscience and eventually completed a PhD in neuroscience as well. He then did a psychiatric residency at the University of Michigan, and he's been a Wolverine ever since. So Jon's major areas of interest are in depression, substance abuse. He's done quite a bit in pain. He uses nuclear medicine techniques in addition to MRI techniques. So he'll be talking today probably quite a bit about SPECT, PET, and a variety of other stuff. So he's, given the nuclear-related work, it's perfect for New Mexico, given that we do everything nuclear here as well. So, without further ado, please join me in welcoming Jon-Kar to New Mexico for his talk entitled "The Placebo Responses: A Resiliency Mechanism."
Thank you very much. Well, it's really a pleasure to be here. It's my first time in New Mexico, and I already have experienced the green chilies for breakfast, and I'm still experiencing them, as a matter of fact, as we are talking. Well, I've been doing this type of work now for a few years. We became interested in placebo responses because, as you know, they are a prominent part of clinical trials. We'll talk a little bit more about that. But where all this work started is trying to understand complex illnesses, things like major depression or bipolar disorder or even substance abuse, and the recognition that these complex illnesses are really the product of interactions between life events and stressors and a genetic background of vulnerability or resiliency, further modifiers such as gender, such as, we know, for example, that women are more predisposed to the development of major depression, but this increased incidence only happens after puberty and not before. So there is something about gonadal maturation that also impacts on the progression and development of depression, as it impacts also substance use, the acquisition and some of the effects of substance use over time. And in many of these illnesses, we do know that there are abnormal stress responses. So we became interested in systems in the brain that are involved in stress responsivity and stress regulation. And one of the ones that we started working with, we've done work on serotonergic systems, dopaminergic. I'll tell you a little bit more about that in a minute. But one of the ones I kind of liked a lot was the opioid system, the endogenous opioid system. And the reason why is because it's phylogenetically a very primitive system. You have these endogenous opioids like pro-opiomelanocortin that are present in the squid, that are involved in stress responses. So, for example, when is the place where the endogenous opioids, and in fact, of course, the opiates will work, but it's also a system that becomes activated under severe levels of stress, but typically in a manner that is compensatory. So you have a stressor. And you've heard about history, stories about people in the battlefield, in the middle of the battle, they get wounded, they don't feel anything. Well, that's stress-induced analgesia, and that's actually mediated through the endogenous opioid system and the opioid receptors. But it's also, being such a primitive system, is involved in a variety of different effects, including affiliative social behavior. So, for example, animals that are nude of the mu opioid receptor, the rat moms, they cannot develop attachment with the pups. So there is an element that relates to attachment behavior that is important. And it seems to be important also in responses to salient and appetitive stimuli, including food and drugs of abuse. That's actually a line of work in the laboratory. And there is all data that actually relates it to placebo effects. And I will tell you that in a little bit in some time. But I want to emphasize one thing, that if you look at the animal literature, the direction of the modulation is typically suppressive. If you release beta-endorphin, enkephalins, the endomorphins, acting on mu opioid receptors, typically when you activate the system, the modulation is suppressive. So if there is stress, it tries to reduce the stress. If there is anxiety, it reduces anxiety. And typically it's activated by stimuli that are actually that threatens the homeostasis of the organism. So brief pain will not activate the system, but if you maintain pain for a while, it will. Things along those lines. So it's an homeostatic mechanism. And in addition, this is a system that is heavily interconnected with another system that is involved in reward mechanisms, which is a dopamine system, as you know well. And there are dopamine-opioid interactions at multiple levels of the neuraxis, including the ventral tegmental area, where mu-opioid receptors modulate the firing of dopaminergic cells. There are also interactions in the nucleus accumbens, in the ventral pallidum, which are thought to be involved in appetitive behavior. And in fact, we know that both dopamine and the opioid system and mu-opioid receptors are integral for the hedonic response to things like sugary drinks in the animal model or appetitive foods in humans and so forth. And both these systems are also thought to be behind the effects of drugs of abuse. So for example, we know that in chronic cocaine abusers, there is a reduction in enkephalin mRNA in the basal ganglia and a compensatory increase in mu-opioid receptor availability in the same regions. And this is something that we actually saw some years ago now in cocaine-dependent volunteers, where we saw an upregulation of mu-opioid receptors in a number of brain regions that were actually associated with craving for the substance. And we've also studied the effect of nicotine, for example, on mu-opioid receptor availability, showing that the acute administration of nicotine actually not only releases dopamine, as we already knew, but also releases endogenous opioids acting on mu-opioid receptors. Again, systems that are both dopaminergic and the opioid quite linked to each other. So how do we go about all this? I mean, you guys are extremely familiar with functional MRI. I know you do a lot of work in the particular area, and because you are so good at it, I wasn't going to tell you anything that you didn't know. So I decided I would tell you something that you probably don't know about very much, which is looking at molecular imaging with positron emission tomography. In my lab, we use this complementarily. So basically, we look at chemistry, as we look at function with BOLD, or structure with DTI and anatomy, resting state, and so forth. And as much as we can, we try to relate one to the other. So we go genes, chemistry, function, behavior kind of thing. So what we do is we take a radio tracer. These actually are images of carbon-11 carfentanil, which is a new opioid receptor radio tracer. It's an agonist, labeled with carbon-11. So you introduce it intravenously, over time becomes incorporated into binding sites. So the areas that are more red, you have more binding, areas that are more blue, you have less binding. And we then take all this information over time, and we do a kinetic analysis, and we then generate parametric maps that tells you something. Typically, what we measure is binding potential, which is the concentration of the receptors or enzymes, divided by their affinity for the radioligand, Bmax over Kd. And those are the images that you actually see there. And those are done on the boxer by boxer basis. Then we do the same work that you do with MR, which is you co-register with anatomy, then you non-linearly standardize, linear standardize to ICBM, MRI coordinates, and use SPM or other statistical packages to look at differences between groups, effects of task, and things along those lines. So a few things that we've done with this radio tracer that are kind of relevant to your work and relevant to some of the work in placebos we've done has been the use of challenges. So not only we look at molecular markers statically, which is what most people do, but one of the things that we've done in the lab, and I hope we've done well, is to use as much as possible physiological challenges that then would change the tone of a particular neurotransmitter system. So for example, in this particular case, this was done in healthy controls, sample of healthy women, where we asked them to recall an event in their lives that induced them profound sadness, and then recall that event over a period of about 30 minutes with, we also had some cues of what those events were, so we reminded them of this sadness state, and the sadness state actually was maintained over a period of half an hour. What we observed was that, in fact, when we did that, there was a deactivation of the opioid system. And we say deactivation because we were seeing more of these receptors available. So when we measure the receptor, you measure availability of receptor in vivo. If you see a release of transmitters, you will see less receptors available for binding. If you see less activity, the receptors can go up. In this case, they went up. Binding potential went up. And the more they went up, the lesser the tone of the endogenous opioid system, the more the negative affect. And this was actually taking place in areas such as the subgenual, pregenual anterior cingulate, the nucleus accumbens, ventral basal ganglia, insular cortex. And the more they deactivated the system, the less the positive affect, effects that were actually localized in the amygdala and in the nucleus accumbens. This was done in healthy controls. And then we went on to look at this in patients diagnosed with major depression. And in that particular sample, we saw large reductions actually in the thalamus, in mu-opioid binding. In a larger sample, we also saw changes in the cingulate. And the more the reduction in this mu-opioid binding, the larger actually the cortisol in plasma, ACTH, which are hormones related to stress. So the greater the elevation in the stress hormones in plasma, the more the reduction in this mu-opioid availability in these brain regions. What was interesting about this is that when we then later on treated these patients with fluoxetine for 12 weeks, and then we looked again, what we noticed was that those that had the largest reductions, they were the more abnormal, if you want, in mu-opioid availability, those actually were the ones that were non-responsive to treatment, to a standard SSRI treatment. So there was something about this stress system that was dysregulated, that was pointing to a poor treatment response. And then when we did the sadness induction, we actually found exactly the opposite of what we found in the controls, which was some of these patients actually activated more than the controls. And the ones that overactivated were actually the ones that were non-responsive. So what this data was telling us is that there are subtypes of depression, that some of these subtypes of depression are related to alteration in systems of the brain, like the opioid or the endocrine system, which are related to an exaggerated stress response. So there was an exaggerated release of these endogenous opioids in response to stress, to the sadness challenge, which we didn't see in healthy controls, nor we see in people who were responsive to treatment, but we did see in people who were not responsive to treatment. So it was identifying some form of phenotype. And now we're actually looking at these effects in relation to potential peripheral biomarkers. So we are looking at pro-inflammatory cytokines, things like IL-1 family cytokines. So we are finding that particularly with the IL-1 family, IL-1 beta, IL-1 receptor antagonist, and IL-18, we are finding that in patients with depression, we are seeing elevations of some of these pro-inflammatory cytokines, and in fact, these elevations are related to mu-opioid receptor neurotransmission in a manner that is actually rather localized in the amygdala. So there is this connection between peripheral inflammatory systems and amygdala function in patients diagnosed with major depression, which is actually becoming rather consistent across samples. And we are following up not just in depression, but also in other conditions as well. Then we've done this type of studies in other samples. We also looked at borderline personality disorder. For those of you who are not familiar with borderline personality disorder, BPD is characterized by emotional instability, irritability, difficulties in interpersonal relationships, self-injurious behavior. So it's a form of emotion dysregulation. It's not major depression. It can be comorbid, but it's different than major depression. And in fact, what we saw in borderline personality disorder was that at baseline, during the neutral condition, there was actually, contrary to what we saw in depression, an upregulation of these mu-opioid receptors. Or maybe there is kind of a decreased baseline tone. But when we did the sadness induction, actually they overactivated in most regions, like the pregenual anterior cingulate, the prefrontal cortex, the pallidum, the amygdala. They overactivated endogenous opioid system. So there was an exaggerated response to this sadness induction protocol, suggesting that what is going on in borderline personality disorder is that there is a dysregulation of the normal regulatory processes that may be mediated through the opioid system in these patients that may contribute to some of their symptoms. And in fact, what we find in this particular sample is that the more they were able to release these endogenous opioids in response to sadness induction, the less severe the symptoms. So those borderline patients who were more able to release endogenous opioids in response to this emotional challenge, those people actually were having less emotionality and less dissociation symptoms during the study. So they were more able to compensate for their emotional dysregulation. Again, goes to the idea that this particular neurotransmitter system is kind of involved in the regulatory processes related to emotion. So you have a given, receptor availability at baseline, which it probably involves the receptors plus some degree of tone. You can deactivate the system. When you deactivate, more of the radioligand goes into the receptor sites. And when you activate it, the radioligand is being displaced, receptors are being either internalized as well potentially, or they go from high affinity to low affinity, they are less available for the radioligand to bind, and you see a reduction in the signal. And this is the type of things that I was showing you a couple of minutes ago. So the other one that I kind of like a lot is the use of, well, we've struggled with the idea of looking at stressors in the lab. We've gone through different experimental paradigms, and one that actually worked really well for us was using pain. Because everybody understands pain. It's a universal stressor, right? I mean, if you are in pain for half an hour, it hurts. Everybody can identify it, everybody can rate it. So it's relatively simple. And the model that we use actually was originally developed by a collaborator of mine, who is a dentist, and it was a temporomandibular pain model. And you simply take a needle, put it into the masseter muscle, and you inject hypertonic saline, 5%, which causes pain. It's low volumes, actually. And then what they do is they rate the pain every 15 seconds. The rating goes into a computer controller, which then changes the infusion rate to maintain it constant over time. The idea is that it's like a block design, if you want, with fMRI. The idea is to maintain the same stimulus constant over time. If you weren't doing that, if you just introduce one single stimulus, the pain will go away right away, or it will become compensated. Anti-nociceptive systems become activated. So that's our pain challenge. It works really well. You turn off the system, and pain goes away. So it's very safe. And then we have a control condition that controls for the non-specific elements of the pain signal. The whole rating thing, and the fact that you have a needle in the face, and that's normal saline, 0.9% saline. And you can randomize them and so forth. So it gives you a fair amount of flexibility. This is how our studies look like. There's a PET scanner. So VAS pain becomes constant over time. And the infusion rate actually increases over time as anti-nociceptive systems become activated. And that infusion rate actually gives you a measure of pain sensitivity. So it's an objective measure of pain sensitivity in the scanner. That's the rating scale that they use, electronic rating scale, and then goes to the computer, and then it changes the infusion. So using these, what we found was that, yes, indeed, you can see the release of endogenous opioids when you maintain pain for about 20 minutes. You cannot also think of pain as something that is exclusively physical. There is an emotional component to pain. When you are in pain, you don't feel just physically bad, you feel emotionally bad. Your emotions go down, your ratings of negative emotion go up, positive emotion goes down. So it is an emotional challenge, not just a physical challenge, especially if you maintain it over time. Brief pain, not so much. And the one thing that we found was that there was tremendous inter-individual variability. So some people had baseline binding that varied from like 1.2 up to 3. Huge differences from subject to subject. Not only that, when you introduce the challenge, although the challenge was rated exactly the same by all subjects, because they were targeting a 40 DAS scale pain intensity, the amount of release of endogenous opioids, meaning the change in binding potential from your baseline to your active condition, varied tremendously from individual to individual. So, for example, this person had a large change, this one very little. And they were kind of all over the place. But this inter-individual variation had a physiological significance. So the more the release of endogenous opioids, the more the suppression of sensory elements of the pain experience as captured by a McGill pain questionnaire in some areas of the brain, such as the basal ganglia, the amygdala, some areas of the cingulate cortex. In some other areas, it was related to the affective or unpleasantness quality of the pain, such as in the thalamus, so the basal ganglia, dorsal cingulate, and in other areas still was related to the affective state of the volunteers while they were undergoing the challenge, as measured with a positive and negative affective schedule. And that was particularly localized in the basal ganglia, in the ventral basal ganglia. So inter-individual variation. And as I told you before, dopaminergic and opioid systems are actually heavily interconnected. Similar things happen with the dopamine system. When you think of dopamine, you think of reward, right? No. You think salience. Okay, dopamine responds to salience, to things that are actually changing your environment, and that can be positive or negative. It can go in both directions. And In fact, when we did the same challenge and we used carbon-11 raclopride, which is a dopamine tracer, you have release of dopamine all over the basal ganglia, both ventral and dorsal, and then we broke it down into the specific experience of pain, subtracting out the non-specific element of our non-painful normal saline stimulus, the release of dopamine in the dorsal areas, the ones that are more related to somatosensory input from the cortex, and was correlated with the McGill-Pain questionnaire sensory and intensity scores exclusively. So it was the sensory element of pain that seemed to be related to dopamine release in a positive fashion. The more dopamine release, the more they rated the pain as being more severe. While in the basal ganglia, actually, was related to the non-specific elements of the pain signal. So we subtracted out the specific, we kept the non-specific element, and in fact, dopamine release in the nucleus accumbens, as you can see here, was linearly correlated with fear and with PANAS negative affect scores. So dorsal basal ganglia, more specific elements of pain, more ventral was the one, the non-specific or more emotional elements of the pain signal. So this led us to looking at different genetic variants. We've done a fair amount of work actually looking at neuropeptide Y and various other genetic variants. The first one we did was catechol orthomethyltransferase. And the hypothesis behind looking at this particular polymorphism is, number one, it was very common. Number two, we know it impacts dopamine and it's been involved in cognition, for example, in schizophrenia and in healthy controls. So, and we knew from the substance abuse literature that if you have too much dopamine around, if you have a situation such as the MET allele of this particular gene, which is associated with less thermostability of the enzyme and a fourfold reduction in catechol orthomethyltransferase activity, meaning that you will have more dopamine, more norepinephrine around. We know that from the substance abuse literature that when you do that, when you have too much dopamine around, that you will have a reduction in enkephalin mRNA, an increase in receptor binding, which we had originally shown was related to craving cocaine dependence. And this affects the prefrontal cortex, the striatopyramidal pathway, and so forth, and this will be the MET allele. And if you have the VAL allele, the ones that metabolizes better the catecholamines, you would expect to have the opposite situation, which is an increase in the opioid peptides and in the reduction in binding, and this would be the VAL allele. And in fact, that's exactly what we found. And individuals that were VAL-VAL homozygotes had the lowest binding, but the greatest capacity to release endogenous opioids to the challenge. These individuals were the ones that experienced the least pain during the challenge. They rated the pain as less severe. They were more pain sensitive, less pain sensitive, and they had less negative emotional states during the challenge. While those that released the least, they were the meta-allele individuals with the heterozygotes in between, were the ones that actually experienced the pain as more severe, were more pain sensitive, and so forth. And later on, this particular gene has actually been associated with risk for the development of some pathological conditions such as temporomandibular pain. In women, they are talking about fibromyalgia as well. So it's one of these risk genes actually out there. And it does have some implications. If you look at fibromyalgia, for example, which is an idiopathic pain condition, we see profound reductions in the opioid receptor availability in these women diagnosed with this disorder, which in fact were associated with clinical pain ratings. So we know there is an alteration in these systems in persistent pain conditions. Fibromyalgia is an interesting one because of its comorbidity with mood disorders, which is about 55%. The other element, which is something that you are interested in, is that we are also looking at these as potential biomarkers. And there is a collaborator of ours, Richard Harris, who is an acupuncturist, and he likes acupuncture, and he wanted to do a study on acupuncture looking at the opioid system because there is this long-standing theory that some of the effects of acupuncture in pain are actually induced by the endogenous opioid system. And he said, great, it's going to be a placebo. This is going to be a really cool study. So this should look like a placebo. Lo and behold, it didn't. So we actually did a study where we did sham versus traditional Chinese acupuncture, and we compared the data between the two. And when we did the sham, there was, if nothing else, a reduction in receptor availability after the needling, the acupuncture needling, which was associated with actually improvement in pain. In fact, true acupuncture, traditional Chinese acupuncture, and some acupuncture were not different from each other in terms of efficacy. They were equally efficacious in relieving pain. They were actually, both of them, better than treatment as usual, which was basically regular treatment in the clinic. So, true acupuncture and traditional Chinese acupuncture and some acupuncture didn't differentiate from each other from a placebo perspective, but they were both different, better actually, than treatment as usual in the clinic. But they were different from each other in the biochemical markers. When we looked at receptor availability, traditional Chinese acupuncture actually increased the availability of receptors both acutely and chronically after 4 weeks of treatment, after 4 weeks of treatment, 3 treatments per week. Restoring the availability of receptors that were actually diminished in patients with fibromyalgia. So this simply to say that although the clinical characteristics and the clinical response was similar between the sham and the active conditions, the biomarker was actually differentiating the two, one from the other. Okay? All right. So, and now we are going to be talking a little bit about placebo effects, which is the title of the study. This is actually true data coming from GSK. Actually, this is a study of drug A, flex dose against placebo and paroxetine, which is an established antidepressant. And on the left side, that study did not differentiate drug A, and for that matter, paroxetine, against placebo. They were identical. On the right side, they actually differentiated. And if you follow the lines, you will see that, so this was a successful trial, that was an unsuccessful trial. The difference between that trial and that trial was not the effect of the active drugs, it was actually the same. It's a line that continues from one to the other. You see it's actually about ballpark the same. What was different was that the placebo effect was smaller in the second trial. So what made the trial successful, and in fact this drug went through FDA approval, was not that the drug was having a better effect than the other trial, is that the placebo effect was smaller. So this led to the company actually screening their sites, looking at the sites that had a more placebo effect, because probably much of this, some of this, has to do with patient accrual and ascertainment biases, how people do these clinical trials out there in the community. However, there is something called a true placebo effect with physiology. And there is a neurobiology to it, and then we'll talk about that in a second. And the idea is that you may have expectations. You are coming into a particular trial or particular treatment with particular positive or negative, as a matter of fact, expectations. In fact, if you read any clinical trial, you will see that people who are being given a placebo will also have side effects to the drug that are similar to the active drug effects in about 15% of individuals. Okay? That happens. That's called a nocebo effect, and we'll see that consistently in the studies. Okay? So, and now we are learning more and more that these expectations do have effects on circuitry and potentially neurochemistry, and may in fact affect psychophysical responses. And you can say, it's like, well, who cares, right? I mean, this is kind of non-specific, no big deal. However, number one is a confounding clinical trial. So it may help us understanding these processes, may help us with treatment stratification, with the stratification of patient to treatment trials. But you may also think about that as a resiliency mechanism. If you receive a substance that is clearly inert and you actually get better, damn, I'd rather take that, than the other stuff. So the question is, can we really affect pathophysiology? And the oldest literature in this area actually comes from the field of pain. In fact, the original data was back from 1978 from John Levine and others, where they were looking at post-surgical patients or experimental pain models, that if you have expectation that you are receiving a drug that is an analgesic, that there is actually a reduction in pain ratings. But you could argue, well, fine, this is biases in responding, obviously a subjective measure, pain is as subjective as our measurements of mood or affective state. So it's not like you have an objective measurement of pain, right? Well, the funny thing about it is that this effect was antagonized by naloxone or naltrexone, which happen to be opioid receptor antagonists. So there is something about the activation of the endogenous opioid system once you are giving somebody the positive expectations that go in with potential analgesic. And originally, the first study was with Thor Weger, who is now, I understand, a collaborator of yours, working out of Colorado, and using fMRI and facing pain. This was when he was at the University of Michigan. Placebo, the typical cream, was associated with reductions in activity of areas like the anterior cingulate, the thalamus, the insula, and so forth. And the anticipation of the placebo administration was also associated with executive areas like the dorsolateral prefrontal cortex. Interestingly enough, the rostral anterior cingulate activation and its relationship with placebo effects, particularly in analgesia, has now been replicated across a number of different studies and paradigms. So that seems to be quite consistently replicated across studies. When you introduce a placebo, there is something that relates to the activation or modification of the function of the anterior cingulate that seems to be related to the response to placebo administration. So we did this with neurochemistry, okay? We did the studies with carbon-11 carfentanil at the beginning, and we gave them standard clinical trials instructions. We told them, look, this agent may be either an inert substance or a compound that enhances the body's ability to counter pain. You may not be able to tell whether it is effective, but we'll be able to tell with our technology. And then we introduced the placebo intravenously every 4 minutes, which was simply 1 ml of saline given to the subject intravenously at the site while when they were looking and they were being told they were being given the compound. And lo and behold, we saw release of endogenous opioids in areas like the dorsolateral prefrontal cortex, the anterior cingulate, the accumbens, the insular cortex, all areas involving cognition, emotion cognition integration, reward responses, and so forth. And a couple of things happened. Number one, there was tremendous inter-individual variability. Some people released very little. Some people released a lot. But there was no particular break between responders and non-responders. It was definitely a continuum. There was completely a continuous response. Nobody could be classified truly as a non-responder versus a responder. There was just there was no particular way of classifying people, other than criteria you may establish yourself. But this inter-individual variability again had significant correlations in the cingulate. The activation of the endogenous opioid transmission and mu-opioid receptors in the anterior cingulate was related to the intensity of pain as measured with the DAS, the unpleasantness of pain, McGill pain questionnaire ratings of the sensory elements of pain, and pain tolerance, almost to a 0.1, almost to a perfect correlation between pain tolerance, which is the volume of hypertonic saline infused to maintain pain over time, which is a hidden measure of pain sensitivity, was completely correlated with anterior cingulate activation of the opioid system. In the insular cortex, similar effects. And in the accumbens, and we see it quite consistently, it was also related to pain intensity, but it was more related also to the affective quality of pain and the affective state of the individuals during the study, the POMS-TMD, the total mood disturbance score of the profile of mood states, which is a measure of affective distress or distress. Interestingly enough, in the dorsolateral prefrontal cortex, the more the activation of the opioid system in the area, the less the anticipated analgesic effect. So it was not related to the pain characteristics, but it was more to the expectations in the negative fashion. If they release a lot of endogenous opioids during the in this particular area, there was less of an anticipation of an analgesic effect, which was kind of interesting thing that we've actually seen later on in other studies. We actually subsequently did a few things. We tweaked a few paradigms, changed some of the infusion parameters, randomized things more, and we actually added raclopride to this. And the reason why we were interested in dopamine and measuring dopamine with placebo is because there is a literature that dates back to about 2001, 2002, where people were looking at patients diagnosed with Parkinson's disease that were being given levodopa, Sinemet, for Parkinson's, and there was a placebo control group, and the group in Vancouver noticed that the people who were being given a placebo actually released dopamine in response to placebo administration, both in dorsal areas of the basal ganglia, which were correlated with motor improvement in Parkinson's, and also in the ventral basal ganglia, which were correlated with the to the anticipated benefit of the Sinemet in these particular patients, which is consistent with the role of the nucleus accumbens in reward expectation. So there is a history of dopaminergic involvement in placebo responses in Parkinson's disease. So we looked at this in chronic pain, and we in the experimental pain model that we use, and in fact we replicated prior data. This is mu-opioid receptor availability changes, so release in the dorsal anterior cingulate, nucleus accumbens, periaqueductal gray, subgenuine anterior cingulate, orbitofrontal cortex for the opioid system. There was The more the release of endogenous opioids, the more the change in the VAS intensity ratings that we acquire. Dopamine was also being released in the basal ganglia, ventral caudate, nucleus accumbens, ventral putamen, and both the dopamine and the opioid system responses were correlated to each other. We kind of knew that. We knew that the dopamine system actually is involving the release of opioids, at least in the striatopalidal pathway. And in fact, these are correlations between the release of dopamine in the nucleus accumbens and the release of opioids in the putamen, amygdala, insular cortex, posterior anterior, as well as in the anterior cingulate, and they were correlated. So it seems like dopamine is probably giving us a go signal, if you want. Hey, look, there is something really cool and salient about this particular placebo I'm getting, and by the way, you now may want to activate your endogenous opioid system in response to this, because this seems to be working for me. So the dopamine kind of gives probably the signal to release these endogenous opioids. And in fact, placebo-induced dopamine release during pain accounted for about 25% of the variance in the formation of placebo analgesic effects. So basically, the dopamine release when you introduce the placebo accounted for about 25% of the variance in the psychophysical effects of the placebo, okay, in these studies, which is quite substantial. So, and then I was telling you about the nocebo effects, right? Well, interesting thing is that a group of these subjects not only don't have a placebo effect, but they have the opposite. If you actually look at individuals that have more than a 20% reduction in pain ratings, that's the green line. But there are individuals, which in the sample was about 15%, 20% of the sample, that they didn't reduce the ratings. In fact, the ratings went up. They had hyperalgesia during the introduction of the placebo with the same instructions. So for whatever reason, these people were just not responding. Not only weren't responding, but they were actually having more pain. And if we actually looked at responders versus non-responders, people who had more of a 20% reduction in the pain ratings versus those who didn't, they were actually differentiated by their capacity to release endogenous opioids and dopamine in response to placebo. The responders release more of these endogenous opioids, and the responders release more dopamine in the ventral basal ganglia. So the nucleus accumbens dopamine and opioid release differentiated responders from non-responders. But they also differentiated placebo versus nocebo responders, which were those individuals that were actually having hyperalgesia in the same regions. And in fact, this is how the data look like. The individuals that were placebo responders release in green more of these endogenous opioids as well as dopamine in the frontal cortex, cingulate, insular cortex, thalamus, accumbens, amygdala. While the nocebo responders reduce the tone of the opioid system respect to the pain challenge in all these regions. So basically, what made a placebo responder or a nocebo responder was that their endogenous opioid system and their dopaminergic system were in opposite directions. Some of them release more, placebo effect, some of them release less, nocebo response. All right. So we said, well, you know, all this is pointing to nucleus accumbens, ventral basal ganglia, dopamine opioid transmission. So I said, well, maybe there is something weird about these people's nucleus accumbens. That, you know, and in fact we did the study. So in this case, actually, we said, let's look at this even without pain. We actually looked at this during pain anticipation. Our studies consist of a saline control, right, where they anticipate they will receive pain, but they actually don't receive pain. They get a 0.9% saline. They don't get the 5% saline. So we looked at that data, where they are actually getting your control state against baseline plus minus placebo administration. And what happened was that we also saw release of dopamine, even when you are anticipating pain, but you don't receive it in the nucleus accumbens. Don't worry about that. It's just something else. And we saw also that when we looked at these individuals and the response of the nucleus accumbens to a standard reward task, the monetary incentive delay task, all you know what this is. You know, it's the presentation of a cue that signals a potential reward, monetary reward of either 20 cents, $1 or $5. The nucleus accumbens responds monotonically, so the lesser response to the lower value, highest response to the higher value. And when we looked at individuals that rated the placebo as more effective than anticipated, or as effective as anticipated, these people had a normal response of the nucleus accumbens to monetary reward expectation. They responded as the usual. The little to little reward, intermediate to intermediate reward, and the largest to the largest reward. However, those people who actually reported that the placebo wasn't working, was less effective, didn't have a normal response to the nucleus accumbens to monetary reward. In fact, it was a flat response as reward increased. So there is something unusual about the encoding of reward value in individuals that are actually placebo non-responders, while the placebo responders, in fact, were, if you want, the healthy ones, the ones that were responding normally to monetary reward. And in fact, nucleus accumbens activity during reward expectation responding predicted about 1/3 of the variance in the formation of placebo analgesic effects. So, kind of an interesting put together of functional and nuclear medicine molecular imaging type of data. So then the question becomes, well, are these mechanisms generalizable, right? I mean, I told you that this data came from Parkinson's disease, where we saw release of dopamine in response to placebo administration when expecting to receive Cinemed. We saw this data looking at dopamine and opioid systems involved in pain, but we also know that these systems, as I told you before, are involved in emotion regulation, are potentially dysregulated in the mood disorders. So we actually have an ongoing study right now looking at the formation of placebo effects in antidepressant trials in patients diagnosed with major depression. What you are seeing here is a sample of 21 patients diagnosed with major depression and medicated. Their Hamilton Depression Rating Scale scores were 17 plus minus 3 or 4, and so it's moderate to severe depression. They were all unmedicated. And we looked at, we did basically something very similar to what we did with pain. We introduced the placebo both orally for 3 days, as well as intravenously in the scanner, and this corresponds to the activation of the endogenous opioid system and the dopaminergic system during the intravenous administration of a placebo with instructions that it was actually an antidepressant compound. Okay? And in fact, we saw similar things to what we saw in pain, which is the activation of the endogenous opioid system in the orbitofrontal cortex, subgenral anterior cingulate, medial prefrontal cortex, as well as the amygdala for the opioid system, and the ventral basal ganglia for the dopamine system. So there are common mechanisms that seem to be underlying the formation of placebo effects across medical conditions. So far we know about Parkinson's, we know about pain, and in this particular case seems to be the case also for major depression. So then the question becomes, what about genetic variation? There is tremendous inter-individual variability in responses to placebo. And this data that is actually now being prepared for publication. It's not quite out yet. We looked at catechol-orthomethyltransferase. Why? Well, we already looked at it before in pain, and it worked. Why not? So we looked at catechol-orthomethyltransferase, and in fact, we saw that the VAL allele, the one that had the better function of the opioid system, also had greater placebo-induced reductions in pain ratings, MPQ total, sensory and pain affect scores from the MPQ, as well as the average VAS intensity when you introduce the placebo. A related enzyme, monoamino oxidase, low function and high function, which is pretty common, actually, we didn't see anything psychophysically. Not much happening there, surprisingly. And the other one that we looked at was the mu-opioid receptor, a common polymorphism, a frequency of about 15% in the general population of the mu-opioid receptor, which is the A118G. And the A allele carriers, the individuals that were homozygous for the wild type, or A type, actually had greater placebo-induced reductions in this case, more the affective quality of the pain, as well as at the affective state of the volunteers, as measured with the profile of mood states, and not so much actually the pain signal itself, more the affect element of pain. In fact, for this, for the catechol-orthomethyltransferase, we saw release of endogenous opioids, as well as dopamine in the ventral basal ganglia, as well as in the dorsal anterior cingulate, which is involved in the regulation of pain, the dorsal frontal cortex, which were correlated with changes in the McGill Pain Questionnaire sensory subscale. While for the mu-opioid receptor, again, we saw both increased release of endogenous opioids and dopamine for individuals that were homozygous for the A allele, more than the G carriers, and those were correlated with changes in the affective subscale of the McGill Pain Questionnaire and in POMS. So there are effects of these common genetic polymorphisms, not only at the level of psychophysics, but also at a level effect of at the level of neurotransmission when you introduce a placebo. So it's a placebo-related gene variation. This is another one that is now being looked at for publication that we looked at, which is the BDNF. You've heard about this particular genetic variant. BDNF, brain-derived neurotrophic factor, is one of the best studied neurotrophic factors, actually. And the MET variant is thought to be associated with a lesser production, lesser concentration of this neurotrophic factor, both in plasma and in brain, and lower effectiveness of BDNF. And what we saw in our studies was that when you look at pain and you actually look at the McGill pain questionnaire in our studies during the pain challenges, we saw that the female MET carriers, but not the male, actually showed higher pain ratings to this challenge than the other individuals. Interestingly enough, when we introduced the placebo, those same MET carriers, the female MET carriers, but not the MET carriers, were the ones that were actually rating placebo effect as being lesser. So all of a sudden they are experiencing more pain, but they are having less placebo effects. And it was only in the females. So it was a sex by gene interaction. And funny enough, this gene was also being associated with two other things that we are interested in, which are vulnerability, and this is measured by the Neo-Personality Inventory revised. And again, the MET allele females were the ones that were having higher vulnerability Neo scores and lower ego resiliency as measured with the rating scale called the ER89, which is it's an ego resiliency type of scale. It speaks of the capacity to withstand stressors and life events. And these individuals that were more vulnerable, rated more pain, had less placebo effect, were also the ones that rated the ER89 lower. They had lower resiliency scores. And when we looked at the pain response, this time looking at dopamine, we looked at dopamine because BDNF and dopamine actually have been linked in animal models. And in fact, we saw that when you introduce the challenge with pain, there is more release of this dopamine in the caudate, as well as in the ventral basal ganglia, in the nucleus accumbens, which was positively and immediately correlated with McGill total scores, with the pain rating scores. And that these effects were actually more prominent in the MET female carriers than in the males or in the Val carriers. So again, we see that at the level of neurotransmission. And when we introduced the placebo, there was a lesser response of dopamine to placebo administration in the nucleus accumbens in the MET carriers. This time we saw it both in males and females compared to the Val carriers. So all of a sudden we seem to be identifying a vulnerability phenotype where you have greater neurotransmitter responses, dopamine, to an stressor in females, MET carriers of the BDNF allele, MET allele of BDNF, and also had lower responses to placebo. In some ways, talking about placebo as a potential resiliency mechanism, these same individuals also were showing lower vulnerability, higher vulnerability scores in the Neo, as well as lower resilience. So that's basically all I want to tell you. I want to tell you about the concept that placebos are not just inert substances, that they are impacting on neurotransmitter systems in the brain, that are important for things like mood, emotion regulation, pain regulation, and that they are, because they have biological underpinnings, that placebo effects actually are also modulated by factors such as gender, you saw that in women, and as well as genes, which then introduce some variance in their formation and their physiological effects. So basically, as we conclude, showing that there is an involvement of the endogenous opioid system in emotion and stress regulation, I show you that, and there is dysregulation of the system in major depression and in borderline personality disorder. We see that both opioid and dopaminergic systems appear involved in the formation of placebo responses, potential across pathologies. We see that in chronic pain. Other people have worked on Parkinson's disease, and now we are seeing that also in major depression. And we are showing that there is inter-individual variation in placebo responses, some of which can be traced to common genetic polymorphisms. And this is relevant not only for clinical trials, but also for the understanding of mechanisms that may be related to vulnerability and resiliency to disease. Again, if you receive a placebo and you get better, I want to be that person. not the one that doesn't get better, and potentially treating responses. And this is actually ongoing work now in a cohort of patients diagnosed with depression that we're actually treating with placebo, with disclosure that is an inactive agent, with deception that is an active agent, and then we treat them with antidepressants, and then we actually see how they do. And the idea would be, I expect to see, although I don't know, I'll let you know next year, whether that people who are actually placebo responders will also be the ones that have the better treatment response, because they are actually more resilient individuals. So that's basically all I wanted to tell you, and acknowledge our group, some of our team, out for dinner and beers, and our funding agencies. Primarily, we get funded through NIDA and National Institute of Mental Health. A little bit of work also through alternative and complementary medicine as well. So, thank you very much. Yeah, not bad.
No, thanks for that.
Okay.
We have time for a few questions. You all on placebo? One.
All right.
Thank you very much for a wonderful presentation. A lot of research that you've summarized.
Oh, this is just a little piece of it.
Yeah, no, I mean, for an hour, you really summarized a lot of really interesting work. Would it be a fair generalization to say that the healthier dopaminergic opioid system is more able sort of to rev up quickly? I mean, it has more room to rev up, and that predicts in general absence of disease, stronger placebo response?
I would expect that. That would be the expectation. And you would expect that those individuals may actually respond better to a placebo administration, while those that have a greater dysfunction, maybe the ones who may respond better to, I don't know, tricyclics or, other drugs that have been around for a long time. That would be the expectation. That's exactly what we are looking for. In fact, we are doing these studies also in patients with chronic pain, where we've demonstrated a dysfunction in these systems, both dopaminergic and opioid, and trying to link the dysfunction of the systems as a consequence of disease, like in depression or in chronic pain, to the development or not development of placebo responses in clinical trials. So.
Andy?
This is going to be a well-formed question, but are you aware of any studies that have looked at, instead of like physical substance, asking the participants to imagine that they're getting a substance or something along those lines to kind of look explicitly more at the expectations?
No. The only studies that have been done with placebo have been with the administration of something. In fact, the majority of people use some form of deception, whereby when they do like pain challenges, they apply a cream and then they surreptitiously reduce the heat that they apply, so the subjects believe that it's actually working. So you do kind of almost a preconditioning kind of process. A lot of people use that kind of paradigm. We don't. We actually don't use any kind of preconditioning because we were interested in inter-individual variation. So we just go in and we tell them, look, you are going to get something, probably will work, maybe not, you tell me, go from there. And so we do create some expectation. Now we've gone to deception. So the way we are doing it now is we actually tell them, look, you are going to get an antidepressant, although they don't. And look, you are going to get a placebo, although they do get the placebo. So we actually do deceive them, and then we debrief them at the end. And so far, so good. Actually works pretty well. So, but you do have to do something to create the expectation. I mean, and you have to do something about it most of the time. The only thing I've seen that is weird is one where somebody told the subjects they were going to be getting a placebo, and they still had a placebo effect. And that was Ted Kapchuk. He did a study with irritable bowel syndrome, which by itself is, a difficult to understand disorder. Nobody really gets it very well. And these people who had irritable bowel syndrome, they were told, look, you are going to get a placebo, and they still got a placebo effect. Go figure. It worked for him. I don't think it works in most conditions. Irritable bowel syndrome maybe, but not in chronic pain. Certainly not major depression, if severe. So.
Yeah.
Yes.
Have you looked at history of a propensity for addiction in some of these predictors?
How do you measure that?
History of drug drug use?
Yeah, no, these are all, the majority of these people that I've shown you data on are healthy controls. There is some use of alcohol primarily, and we are looking at that. As a matter of fact, we are looking at this. We know that this is work actually done by Nora Volkov and others in her lab, where we know that people who have a history of substance abuse or are current substance users have a lesser duration of placebo responses. Drug addicts are very good at identifying their drug of choice. So if they receive a placebo, they may actually have a response of dopamine. In fact, it was shown they have dopaminergic response. But that dopaminergic response goes away like really fast because they realize that, hey, this is not working for me. So they are very good at discriminating. And in fact, the placebo response, as you know, in clinical trials for substance abuse is very, very low. Do you know of another syndrome that has a low placebo response in psychiatry?
OCD?
Yes. And the idea, I mean, I don't know, this hasn't been studied really at neurobiological level, but it's thought that this syndrome, OCD, goes with the reverberation of circuitry, alteration in the functional or with the frontal cortex and basal ganglia networks. And those are the ones that are involved in the formation of placebo effects. So if those systems are compromised, you may actually have, neurobiologically, a low probability of developing placebo responses. What about neurodegenerative disorders, Parkinson's? You would think it has a low placebo effect, right? It actually has a very large placebo effect, which is not very well understood. But if you know what happens in Parkinson's is that by the time you get diagnosed with Parkinson's, 85% of your dopaminergic cells are decreased, are dead, are degenerated. But the rest of them are working like crazy. So any small changes in signal are going to give you a huge effect, simply because if there is just an increase in firing, because of placebo administration, they will give you a big physiological effect, and that's probably what's going on. What about the dementias? The dementias actually have a very low placebo effect. You don't form the cognition necessary to actually provide with the expectation and the updated expectations. Stefan.
I have a more technical question.
Yeah.
In fMRI, we learned that systems like the amygdala are very time dynamic in terms of the regulation, so they habituate very rapidly. Is there any correspondence in the receptor systems that they also experience this time dynamic kind of response, even though I do understand that it's hard to measure this?
Yeah, it's hard to measure that. Probably. I mean, we do know that the opioid system can become desensitized over time. Receptor sites go from high affinity to low affinity after exposure to the endogenous ligand. They go through recycling. Yes, you can do desensitization of some of these systems relatively quickly. Hard to measure with PET, but that's actually one of the reasons why our studies typically maintain a stimulus over a prolonged period of time to be able to get these signals. The temporal resolution for us for PET is much lower than for fMRI because we need to quantify the receptors. So to quantify them, you need data over time, so you do kinetic analysis. And if you want to do release, which then basically implies measuring the receptor again after you do a challenge, you'll need at least challenges that last for about 20 minutes. So they are rather prolonged. But our stimuli are hammers. I mean, we use pain, we use sadness induction, things that are a lot less subtle than most of the things that you use in fMRI. One that has been successful recently has been social rejection. We use like a dating game, and people get rejected online by a lot of people. And this is kind of actually people really respond negatively to that. They also get accepted, so that's good. But and man, that has a huge effect on the systems.
So John, I have a question. So do you think that the placebo effect is related to any personality dimensions? For example, like, they always say that some people are more suggestible than others. And you mentioned you showed a little bit of neo and other types of things.
It doesn't have I didn't want to get into this because I thought it would go too long, but yeah, it does. Let me show you some of the data. This is from the placebo analgesia studies. We looked at 15 trait variables, and we looked all over the place. We used data reduction techniques. So we used the ER89, which is ego resiliency, the 5 domains of the neo, the BISBAS, behavioral inhibition activation scale, LOT-R, which is a measure of optimism, well-being scale, which are trait well-being, psychological, emotional, and social, and the STAI, which is the trait anxiety. 3 variables, when we looked at all of them and the proportion of the variance in placebo and analgesic responses they were predicting, 3 variables, the ER89, the ego resiliency, agreeableness, and eroticism will explain about 1/3 of the variance in placebo analgesia, which is quite considerable. Then we decompose the neofacets into the neofacets agreeable and eroticism into the 12 subscales, and then we reduce the data again. And we came up with 6 scales at the end of the whole thing, which was 4 positive predictors, which were the ER89, neo-altruism, straightforwardness, compliance, as being positive predictors, and 2 negative predictors, self-consciousness and angry hostility. So I guess your patients, your antisocials, will never respond to placebo.
Well, so that's interesting, because they're the opposite of OCD. And so if you said OCD has no responsiveness to placebo, then psychopaths who are the complete opposite.
But they don't have emotional responses to anything.
Well, so that's exactly what I was going to say. There's like an intersection, right? So what you have is, on one dimension, you may have the rumination kind of processes and stuff like that, and psychopaths are down here. But on the other dimension, you have the affective, related stuff in there. So those two things might cancel.
I think so.
Well, because what you're getting, that's what I was getting at, is that these positive and negative predictors, you might see that if you're high loaded on both of those, you would be down there, but if you're, okay.
That would be a good question, actually, to ask. I don't know what is on the study. But if you think about it, actually what makes a good patient, makes a good placebo responder. You're altruistic, you're straightforward, you're compliant, but you are self-conscious and you're angry, you don't respond. And you have low resiliency. So
that's not psychopathic. That's opposite.
Exactly.
Yeah.
So that explains about 1/3 of the formation of placebo analgesic effects.
Sure.
We're also looking at neuropsych testing data.
So this is a very general question about, I'm curious about your advice in terms of grant writing with the new RDocs criteria. You're familiar with the RDocs
Of course,
criteria.
yes.
And I'm going to try to explain why I'm bringing this up. I mean, so your presentation is beautiful in that you show sort of very similar paradigms, PET paradigms, using looking at opiates, looking at dopamine, across many different psychiatric and non-psychiatric conditions. So the presentation is sort of a poster for RDocs, yeah? But on the other hand, RDocs, I mean, this is a whole program of research that you've developed over many, many years.
Right.
You started looking at depression, yeah, and saying with these paradigms, depression is clearly heterogeneous.
Clearly.
So if you're writing a grant following RDocs or keeping RDocs in mind, and you're interested in understanding the heterogeneity of depression, and you're using these very expensive approaches, PET, you can't scan 100 subjects. I mean, how are you going to navigate, this new RDoc system in terms of your approach? I mean, do you think it's going to be helpful in terms of getting grants, or is it going to be problematic because you might want to just say, no, I want to look at depression and understand the heterogeneity of depression, which is essentially not compatible with RDoc?
Right. Well, RDoc really tries to break down disease processes into particular domains that are easier to study. Fear responsiveness, you name it, attentional components, placebo response. That could be an example. Non-response versus response, high response versus low response, things along those lines. You could argue that most of these tap into a stress response and stress response regulation. Whether you look at it with central neurotransmission, or you can look at it with fMRI, or you can look at it with peripheral measures, like cortisol and ACTH, we are probably tapping into a phenotype that is more related to the capacity to regulate stress appropriately. That's probably what all these, at least the opioid system is getting into, is the appropriate regulation of the stress response, as this being a system that is involved in that, in a very primitive way. Dopaminergic system probably, forms part of that. So is it going to be helpful? I mean, I think it's obviously hard to scan everybody with these type of technologies, but you can use other more proximal measures or more distal measures. That's why we are interested in, like, the cytokines, for example. Are they giving us a signal that we can then learn about? That's why we are interested in things like trait variables. Are trait variables going to be correlated with central neurotransmission, with bold responses, with whatever, that then can give us some predictors that can be used in clinical trials in the field. But with